Things about the ear. First is that it does hearing, but also does our equilibrium senses. So let's give it a little background to start. Hearing is going to be part of the ear called the cochlea, which is Latin for snail because it spirals around, looks like a snail kind of. But there's also what we call the equilibrium senses. And this is going to be, there's going to be the semicircular canals that are going to do what we call rotational or dynamic equilibrium. Which way is your head turning? We talked a little bit about this when we were doing the reflex lab, talking about how nystagmus, like you can shut your eyes, but you still know your head is turning up or down or side to side or rolling back and forth. So semicircular canals are telling your brain information about the rotational or dynamic equilibrium of your head. And then there's also going to be what we call the saccule and the utricle. And these are going to do what is called static equilibrium. This is basically the direction of gravity. So we'll spend time looking at hearing proper, like sound, converting sound into electrical signals. But we're also going to be looking at these equilibrium senses, which are also part of the inner ear, the semicircular canals, which convert this rotation of your head into electrical signals for your brain, or the saccule and the utricle, which convert the direction of gravity with respect to your head into an electrical signal for your brain. They all rely on the same basic little cell called the hair cell, just connected up in different ways to do their jobs. So we need to, these all rely on the hair cell. So let us introduce the hair cell. So the hair cell is a mechanoreceptor. It gets its name because it has cilia and stereocilia up on its apical surface. It has one true cilium called the kinocilium. The other ones are actually stereocilia, meaning they look like cilia, but they're actually like overgrown microvilli. They don't actually have microtubules in them. And these are all connected with these like little tip links that are connected up into these channels that can get pulled, that open and close. And basically what happens, if you bend the hair bundle towards the kinocilium, it will depolarize the hair cell. And if you bend it the other way, you hyperpolarize, it becomes more negative. And typically these are connected up to some neuron. And so basically, as you bend the hair bundle towards the kinocilium, this thing becomes more positive and you start activating, you get more action potentials going off towards the brain. When you bend it this way, you bend it away from the kinocilium, you're going to have less voltage. You're going to have less likely to be sending action potentials off towards the brain. So this is at the core of hearing. It's at the core of the equilibrium senses. This little hair cell, depending how you hook it up, you can make it detect the vibration of your eardrum. You can make it detect the rotation of your head. But at the core, it's just if you bend the hair bundle one way, it becomes more positive. If you bend the hair bundle the other way, it becomes more negative. 9 a.m. And that's going to be at the core of the transduction. So we now need to back up and think about the ear in the big picture of your head. So usually you think of the ear, you don't really think much beyond, we have what's called the outer ear, or the auricle. And then we often think about the ear canal, because that's where you put your Q-tips to try to get the earwax out. But the real meat and potatoes of your ear goes beyond that. So this is like the outer ear. Most of the interesting stuff of the ear is in the skull. So this is your skull. We're going to have the middle ear. We're going to have the inner ear. And I'll draw this in much better pictures in a second. But all this stuff that I'm going to be talking about is within the skull bone. And when it's all said and done, we're then going to have the nerve that, the vestibulocochlear nerve that then goes to the brain. But the middle ear and the inner ear are basically like little caves within the skull bone, the temporal bone of your skull. This is going to be, I think, easier if I use some PowerPoint pictures instead of drawing them, because you can see my pictures here are not beautiful. Okay. All right, so this is just showing this idea of the hair cell, right? You take the hair cell, you bend it one way, it becomes depolarized, more positive, releases more neurotransmitter and more likely to be sending action potentials to your target. You bend it the other way, it becomes more negative, you stop releasing neurotransmitter, you don't have the action potentials. So again, this is going to be at the core of the transduction. Hold on a sec. Wait, where's my? Actually, I'm gonna use these are actually pictures from an anatomy book, because anatomy books actually tend to draw this stuff more pretty. So this is kind of a picture of what I've been drawing. Maybe I'll add this into this is from the Martini anatomy book. But it's your book has similar pictures in it as well. So things that we're going to pay attention here. One is the outer ear, which we just talked about. That's basically where the sound is coming in. It's kind of like an ear horn, like those old pictures, like, what's that, Sonny? And they have like that little like funnel to try to like collect the sound. That's kind of what your ear is doing there. When it enters, here's the middle, this is that middle ear here. You can see it, they say middle ear. The middle ear is going to be this little conversion where the air vibration is going to ultimately get converted into a fluid vibration for the inner ear. The inner ear is where the actual phototransduction happens. This is where the hair cells are. The hair cells are in here. But there, all of that stuff is in fluid. It's in, and we're going to talk endolymph and perilymph. If you try to just send the vibration from the air directly into the inner ear where the hair cells are, most of the energy would just bounce back. The middle ear has these little bones, the incus, the stapes, the malleus, also known as the hammer, the anvil, and the stirrup. And they are basically helping efficiently transmit the vibration of the eardrum, the tympanic membrane. So we can efficiently deliver that vibration into the fluid-filled world of the inner ear. So a big part of what the middle ear is doing is providing this ability to efficiently transfer the vibration from the air vibration into a fluid vibration inside the inner ear. When something is wrong with your middle ear, that's gonna create what we call conduction deafness. We're gonna see that as part of our lab today. Like one way to be deaf is just the sound never makes it to the inner ear where the action is happening, right? The middle ear doesn't actually convert sound into electricity, but the inner ear is where we actually convert sound into a vibration into electricity. But if the sound never gets there, you're still deaf. So this idea of conduction deafness is that we're going to look at in lab is if we have some trouble actually getting the vibration into here. That can happen because these bones are ossified and don't vibrate with respect to each other anymore. It could be that there's some bad infection and this is all filled with fluid or something and things don't work anymore. Or it could be your eardrum is punctured, right? The eardrum is kind of like a little drum head that's vibrating in response to the air vibration from sound. So all of those things can cause this conduction deafness that we're going to see in lab. Other things about the middle ear. I'm going to clear off. Again, middle ear. If we have really loud sounds and we actually want to help protect the delicate inner ear from damage from too much vibration, you actually have little muscles that can tighten up and reduce the motion of these little ear bones, these little ossicles. And these are we call collectively are the ossicles. So typically the air vibration comes in here. The eardrum is basically this little membrane, is connective tissue and epithelial on this side. This vibrates. As this vibrates, it vibrates these little bones, which act as a little lever system to efficiently transfer the signal into the inner ear through what we call the oval window here. If the sounds are really loud, we have these little muscles. There's like the tensor tympanum and stapedius. For this class, you don't need to know, you don't have to memorize their name, but you should know that there are little muscles in your middle ear that can tighten up and reduce the transmission of vibration if it's really loud. It's one of the ways you can protect your inner ear. If you remember when we talked about REM, I was saying in REM, all your muscles are paralyzed except for, those eye muscles, as well as some muscles in your middle ear. Those are the ones that can wiggle around. Actually, when you're dreaming, you can actually have those muscles wiggling around as well, that are kind of tensing and relaxing on your little ossicles. Obviously, those are going to be harder to see because they're inside your inner ear. I mean, in your middle ear. I take that back. They're in your middle ear. The other thing we need to talk about the middle ear is, it's filled with air, but it's kind of closed off. One side is walled off with the tympanic membrane, the eardrum. The other side is walled off with the inner ear here. So the air pressure in here has to be equal to the air pressure outside, or else you're going to have unequal pressure across the two sides of the eardrum. Minimally, that's going to be painful. Worst case, you're going to pop your eardrum. It's going to explode. And the air outside changes, right? If you are going up to higher altitudes, the air pressure is lower. Or you go up in an airplane, or if you're diving down to the bottom of a swimming pool, the pressure is getting much more strong. If you're 30 feet down, the air pressure, it's not air pressure, it's water pressure, but the pressure is now twice what it was at the surface. So as the pressure is either higher or lower on this side of the eardrum, we need to make sure we can equalize the other side, or else your eardrum is going to pop. So how do we do that?
Can you release air?
So we actually have a little tube, the eustachian tube, which connects from the middle ear and to the pharynx. What is the pharynx? It's the fancy
Isn't
word.
that like part of your throat?
It is. It's the fancy anatomical word for throat, right? Your laryngopharynx, your oropharynx, nasopharynx. It's just a fancy word for throat. So it opens up into a little opening in the back of your throat, kind of up the upper part in the nasopharynx there. There's a little hole connecting the back of your throat to your ear. You can feel it. You can do the Valsalva maneuver. In fact, if you dive, you got to do this anyway. If you don't do this, your ears are going to pop. If you hold your nose and you blow gently, you can feel your eardrums pooch out. Do you all feel that? You're basically forcing air up the eustachian tube into the middle ear and pushing out your eardrums. When you swallow, it helps expand the eustachian tube and open it up to allow the air pressure to equalize. Whatever the air pressure is in your mouth will become the air pressure within your middle ear. Right? That's why, often people chew gum when they're driving up to Tahoe or something, because every time you swallow, you're helping open up the eustachian tube and equalize the middle ear pressure as the pressure outside keeps becoming less and less. It's also why if you have a bad cold and there's inflammation, so the eustachian tube has trouble opening, it can be really painful to try to fly. Or if you dive, you might not even you have to cancel your scuba dive if you have a lot of inflammation, because if you're not able to equalize the pressure, your eardrums are going to implode as the pressure gets stronger as you dive deeper. So you have to have a good functioning eustachian tube, or else you're going to run into problems with building up excess or not enough pressure in the middle ear. Right? It's people get exploded eardrums on airplane flights sometimes if their eustachian tube is clogged up from a really bad cold or allergies or something. And as the pressure gets less outside and the pressure inside their middle ear can't equalize. If there's too much pressure, you pop the eardrum. You know, the eardrum can heal. It's a tissue that can heal. You can also have, like, you know, middle ear infections and stuff where they actually stick a little tube in through your eardrum in order to drain out the goo that's happening within your middle ear. So anyway, let's go back to annotate. So questions about the middle ear. Middle ear is important because it's got the little ossicles which help efficiently transfer the vibration into the inner ear, which is where the action happens, the transduction happens.
So I have a question about like the pressure. Because I was curious how I was told that babies have a harder time like equalizing the pressure, like if you're on a plane or something like that. I don't know if that's true or not, or why that would be.
So that I don't know. It could be if they're, I mean, a lot of times things in babies just don't work as well because things are still developing. It could be from that, or it could be they're more likely to have some kind of ear infection or something, which would also make it harder to equalize if there's inflammation. But I would, yeah, that's something I think a pediatrician would be able to answer more
Okay.
than I could.
I think it just has to do with they can't like feel the pressure, or they can't do anything about it, like where we feel the pressure in our ears and we can kind of, you know, pop them and equalize them more than anything, because they suggest having your like giving a baby a pacifier or something, because that sucking motion will help them
Right.
clear the air pressure.
Right. So that would be similar to like giving more of a grown-up the gum to chew and swallow. So, or I guess if you could give a pacifier to an adult, but yeah. So now we're gonna get to the inner ear.
I just have one really quick question. So the air vibration comes from the external ear into this middle ear, and then the fluid vibration happens in the inner ear?
Yes.
Okay.
So we have, you know, so like when I am talking, like if you were in front of me, it would be more direct. Instead, it's through an electrical signal that then becomes your speaker. But regardless, the speaker in front of your computer is literally a diaphragm, which is going back and forth and creating these compression waves in the air. And then those compression waves are propagating. And when they hit the eardrum, they make the eardrum vibrate in a sympathetic pattern to the compression waves in the air that were originally formed by my vocal cords. So, whoops. Hold on, I need to stop. Clear, stop. So by having the eardrum going back and forth from the air vibration, that creates the kind of push-pull in the ossicles, the middle ear. That is going to now transmit that same vibrational pattern into the fluid of the inner ear through this oval window, this little membrane that is getting kind of plungerized by the stirrup, by the stapes bone there. So that's where we're going to look now. Let me get rid of my annotations. Okay. This is kind of showing. This is what compression waves look like. These are representing actual like little air molecules, you know, that then go in and they basically kind of impact your eardrum. This is kind of showing this idea. You know, we're going to see the inner ear is much more than just the cochlea. The cochlea is the part doing hearing, but this is kind of showing the sound waves coming in from the outside, vibrating the eardrum, the tympanic membrane, which is then working on this lever system of the ossicles, transmitting into the fluid world of the inner ear. When we get to the inner ear, we're going to see the hair cells are located here in the cochlear duct. So as the things wiggle around in here, it's going to wiggle the hair cells, which is going to create the electrical signal that you perceive as sound. If you do engineering stuff, this idea here is impedance matching with the inner ear. If you take just the air vibration and try to bounce it straight off of the oval window, most of the energy would just reflect back rather than be transmitted into the inner ear. So having the system of ossicles allows you to make sure most of that energy is going into the inner ear rather than just bouncing off and not being sensed. Okay. So the inner ear is lots of different parts. Oh my God, we're running kind of low on time here. But it's all going to be good. So here's that outer ear, the middle ear, the inner ear. So the inner ear has this cochlea, which looks like a snail. It's got these vestibular things. Hold on, I want to find a better picture here. This is more what it looks like. All the different parts here. So we have this vestibular part. This has the semicircular canals and the utricle and the saccule. It also has the cochlea, which is the part that's going to be doing the sound. So let's start with the vestibular stuff, and then we'll finish with the cochlea. So this is this part of the inner ear that I'm going to talk about has nothing to do with sound, has nothing to do with the eardrum or the ossicles. This is just always happening in your inner ear, inside your skull bone. So all this other stuff I've been talking about is going to come into play when we get to the cochlea and sound. But let's start with the parts that aren't sound. Here in this part of the inner ear, we have what are called the utricle and the saccule, which are basically hair cells which wiggle. And if they move back and forth, we get voltage changes. And then they have this gelatinous membrane full of what are called otoliths. These are otoliths are basically calcium stones. They're little calcium carbonate stones. So we basically have hair cells with a bunch of rocks on top. So imagine inside your skull right now, in fact, you have hair cells with a bunch of rocks on top, and gravity pulls on the rocks. So if you move your head in some direction like this, gravity is going to pull on those otoliths. Those otoliths are going to get pulled with respect to gravity. They're going to bend those little hair cells. When those hair cells bend, what happens?
Action potentials.
Yeah, we're going to send action potentials to the brain, which is going to then know, oh, gravity is pulling in a different direction, right? So the two the saccule and the utricle, they're the same thing, just in different orientations. So you can tell gravity in different orientations with respect to your head. But in both cases, there's going to be hair cells and some kind of gelatinous membrane, and then those little calcium carbonate rocks, the otoliths. Otoliths means ear stones. And like I said, when you tilt your head, that is gonna change the direction of gravity on those stones, which is then gonna create a bending on these hair bundles, which is then gonna send an electrical message to your brain that the orientation of gravity with respect to your head is now this instead of this instead of that. Right, so if your eyes are closed even, you have information about which direction is gravity pulling, because as gravity pulls on these otoliths, which are anchored into your inner ear, you know the direction of gravity based on the bending of the hair cells. This little patch of hair cells with otoliths, they call them macula. It just means spot. There's a saccular macula and a utricular macula. It just means that little patch of hair cells in that inner ear that are sitting there with the little gelatinous membrane and otoliths. So we call this static equilibrium. Because it's, even if you're not moving, it's giving you information about which way is gravity pulling. Sometimes they call this also linear acceleration, if we want to be complete. Right, because there's, think about general relativity, right? There's no difference between sitting on Earth with gravity pulling down at 9.8 meters per second squared, or being in a Tesla accelerating at that speed and getting plastered onto the back of the seat. It's just, linear acceleration and gravity, are the same thing. Gravity is linear acceleration down towards the center of the Earth. So if you're getting plastered on the back of your car as you're accelerating and you feel that you'd also be activating your saccule and your utricle. But anyway, so that's static equilibrium. The other kind of equilibrium that's happening in the inner ear is rotational equilibrium. That is happening in the semicircular canals. They look kind of like these Mickey Mouse ears coming up off of the inner ear here. And those are very different. Here, those have hair cells as well, but the hair cells are connected to this kind of gelatinous curtain called the cupula. It'll be better if I actually draw it. So I'm going to draw one, but there's three of them for the three different axes that your head can rotate, right? Your head can rotate. If you were an airplane, you could do the yaw, the pitch, and the roll, right? So there's three different dimensions, three different axes that you can rotate in. You have one of these for each of the axes. Again, they're embedded within your skull bones. So as you move your head, this whole thing moves as a unit. You have a canal. You have this swelling called the ampulla, and then we have hair cells. And the hair cells have a little gelatinous membrane called the cupula. Here are my little hair cells. And then this whole system is filled with fluid, the endolymph. And then the other thing, just to kind of remember, this is your skull. So when you move your skull, you're moving this whole, if I'm rotating my skull, like, like this whole system is moving. Except the fluid. What happens if I take a glass of water and I start moving the glass of water, rotating it back and forth? Does the water just move along with the glass?
No.
It has inertia, right? Every people, inertia, the classic, right? If I have blah, 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 blah. Right, the classic thing, if I have a piece of paper here and I have this thing here, and I pull this fast, the stapler didn't move, right? Inertia. So if my skull is moving back and forth, this fluid actually does not move along. The fluid stays still, which means it's going to push. Let me get the right, like when, let's say my head was going this way. The fluid isn't moving. The fluid is kind of staying still as my head goes that way. It's going to create a pressure on this little gelatinous membrane, bending the hair cells backwards, whichever. So like it might be good to just get out of Zoom for a sec. I mean, think about air. Air is a fluid, right? Air is, you don't see it so well, but like if let me get rid of this silly virtual background so you can see this, right? If I have some membrane and I move it this way, but the air isn't really moving, then the membrane gets pushed the opposite way, right? Whichever way I'm going, the membrane is kind of getting pushed by the air that's not moving. So this is what's happening, like in your circular, semicircular canal in your middle ear, in your inner ear. When you twist your head one way, the little cupula bends the opposite way and bends the hair cells, and your brain's getting information that you just turned your head in that way or that way. And again, you have them in the different axes. So I can tell if I'm nodding or if I'm going, no, 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 if I'm going, blue, 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 whatever. So does that make sense? That was what they would call dynamic equilibrium, because it's not telling you information unless you're actually moving your head. It's telling you which way is my head rotating. It's also called rotational acceleration. Why do people get dizzy if they just keep like spinning around and around and around and around and around and around and around?
Because the fluid inside the canals is still moving even after you stop, and then you have that weird sense of you have stopped, but you feel like you're still moving.
Yeah, so exactly. So if you keep spinning and spinning, eventually the fluid is turning along with your head. So if you've ever just spun around and around, you actually aren't that dizzy while you're actually spinning. In fact, you're not dizzy at all while you're spinning. You're dizzy when you stop spinning, because then the fluid is going to keep trying to move because it's got inertia, but now the inertia is to keep spinning and your head is not moving. So it's going to be pushing against the little cupula, and your brain is going to think you're spinning when you're not. And that's going to be the sense of being dizzy, a vertigo, right?
So the fluid's it's not actually moving. It's just maintaining that pressure.
Yeah, it's trying to move. It can't actually move through, but it keeps trying to get through, right? So if I'm in, how, if I wanted to, let's say I'm a ballet dancer and I want to do my spins, but not like fall on my face after I've done my pretty pirouettes, how do I actually get around that?
This you spot.
You do spotting, right? So if you're spinning your body, but you don't let your head move, and then you just kind of snap your head really fast. So that is going to prevent the fluid from getting entrained and moving along with your, if you're just kind of doing like that, by the time you finally are done spinning, the fluid never started spinning along with your head. So when you stop with your pirouette, you're not going to be dizzy. Whereas if you just were spinning around like this constantly, eventually the fluids and the head are all moving together, and then I stop and now I am dizzy. And then if you look at my eyes, it'd be, what's the word for the flicking of the eyes when you've got vertigo?
Nystagmus.
Yeah, nystagmus. So that is rotational or dynamic equilibrium. So let me give it a name. So rotational acceleration, dynamic equilibrium. These are our three semicircular canals. If there are things that affect the pressure in there of those fluids, you can get very debilitating vertigo, where you just always feel kind of dizzy. Something, in fact, I should give you some names here. Hold on. Let me share. I mean, I'm not getting into a lot of the detail because we're not in anatomy, but you have like the bony labyrinth, the membranous labyrinth. So these are all called kind of the labyrinth because it's all this big crazy network of tunnels inside your skull bone. So like there's a condition called labyrinthitis, inflammation of the labyrinth. And one of the symptoms is just feeling car sick all the time, basically. It's like you feel like you're moving in some ways, but you're not moving. There's a disconnect between what you're experiencing and what your senses are telling you you are experiencing. And yeah, this can happen like if people get ear infections and stuff where, you just want to vomit because you just constantly feel like there's some disconnect where you're spinning when you don't see you're spinning and things like that. So there's another number of problems that can happen. It can happen from, again, ear infections, or sometimes people don't even know where it's coming from, but it can be quite debilitating if there is signals going to your brain that your head is turning when in fact you're not.
What about alcohol-induced spinning? Is it messing with this?
That I'm not sure the mechanism of that. Yeah, I do. So the spins, yeah, I don't know exactly what causes the spins. Yeah, you have to be pretty darn drunk for that to happen. Yeah, I mean, that happened in college. That has not happened in a while. I don't know what happens there exactly, what the mechanism is. So labyrinth. This part of the inner ear is called the vestibule officially, vestibule like the little entry. So often these equilibrium senses, both the saccule, the utricle, and the semicircular canals, they call them like vestibular senses. So often vestibular senses, equilibrium senses kind of mean the same thing. And those are the parts of the ear that are not about hearing. They're about, your balance, direction of gravity, which direction is your head moving in space. Any questions about these aspects of the ear? Okay, so we still have to finish the ear, but we will be able to do that. And then in our lab, the lab is going to be some fun stuff about the ear as well. But all we have left is the cochlea, really, which is the most complicated part, but it's not that complicated. Okay. So the cochlea. Latin for snail. It's basically got like these 2.5 turns. So if we kind of come back to the outer ear, there's the tympanic membrane, the eardrum. There's the middle ear. And that feeds into the inner ear. And again, the inner ear has the semicircular canals that we've been talking about. It has the saccule and the utricle here in the vestibule. And then it has this big snail-like thing sitting on the bottom there. That's where the special sense of hearing is going to happen in that part. And again, reminder to you all, that all of this stuff is in the skull. So what we're going to do now is think about sound, again, which we've been talking about, coming in as sound waves in the outer ear, vibrating those ossicles, hammer, anvil, and the stirrup. And now we're going to look at this in a little more detail. That little place where the ossicles push and pull on the inner ear is called the oval window. Make it maybe black, because that would be, you would be able to see it. Its window makes it sound like it's an opening, but it's an opening covered with a membrane though. So it's a membrane that then these little bones can push and pull on. So as you push and pull on this oval window, you're creating basically pressure waves in the fluid inside the inner ear. Because fluid is not compressible, or not very compressible, I should say, if you push on the oval window, it's not going to move unless something can give. So there's another little window called the round window. That basically is kind of the partner. Like if you push on the oval window, the round window will pooch out a bit. If you pull on the oval window, the round window will suck in a little bit. So, they just kind of do the opposite things of each other to allow the vibrations to exist in the fluid. So that is kind of the connection between the middle ear and the inner ear. The oval window is how the vibration enters into the inner ear from the ossicles. And then the last part, we need to add in a little anatomy. I've talked about there being bone, that this is all in the middle of. The cave is called the bony labyrinth. And then there is the all the membranous stuff inside, and all that membranous stuff they call the membranous labyrinth in here. And between the membranous labyrinth and the bony labyrinth, you get different little like tunnels where you have different kinds of fluid. So you can have like fluid in between the bone and the membrane, or fluid inside tubes in the membrane. So for the purposes of this class, don't worry about all of the details of the anatomy. Mainly what we're going to talk about are 3 ducts that end up like this snail that we are seeing here with 3 or 2.5 turns of the snail, because there's also this membranous labyrinth inside going up the snail. It actually creates 3 tunnels. There's this tunnel from the bone to the membrane, the tunnel inside the membrane, and then the tunnel on the other side of the membrane to the bone again. So we have what we're going to see. They're going to be called the, it's going to be the vestibular duct, the tympanic duct, and the cochlear duct. I'm going to draw them all much prettier, but just kind of give you the sense that the vibration from the air to the eardrum, to the ossicles, to the oval window are then going to create these fluid waves that are going to travel up the vestibular duct, around the very tip, and then all the way out the tympanic duct, and then decompress out on the round window. So that's what we're going to be looking at now, is this kind of flow of vibration up and down the snail. So let's take a look at the vibration up and down the snail. Blah, 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 blah. Here we go. Let me go into my share screen.
Mr. Egert, really fast, I will be able to do the test on Thursday.
Oh, awesome. Thank you. So I will send out a thing as soon as we're done here. Okay, so this is showing the snail, and you can see the snail has different canals in it. Back. So if you take the snail and you unwind the snail, so that's what this picture, it's way easier to understand the cochlea instead of thinking about it being all these spirals. You can think about the oval window as being the entrance into this, what we call the tympanic duct, the vestibular duct, because it's kind of where the vibration enters. Vestibule is kind of an entrance. Helicotrema, this is the tip of the snail. This would be, here's the cochlea, the very tip of the snail is the helicotrema. So basically you're spiraling up to the tip of the snail, and now this tympanic duct is spiraling back down, out. Here's the round window. So vibration, again, comes in, ear canal, vibrates eardrum, vibrates ossicles, vibrates oval window. Oval window then sends the vibration up this duct in the cochlea called the vestibular duct until we get to the helicotrema. And then it returns back and the vibration can decompress here on the round window. This duct in between is called the cochlear duct. It's where we find the organ of Corti. The organ of Corti has the hair cells.
Do you want us to know each of these structures, or is this just for reference?
You should know that it goes up the vestibular duct and out the tympanic duct. The thing that's going to be really important is the basilar membrane and the organ of Corti, because those are the ones that are actually going to be involved with the real transformation of vibration into electrical signal. Like I said, we are concerned a lot with the sensory transduction, and the hair cells do the transduction. They convert the movement into electrical signal, and they're in this organ of Corti. sitting on top of the basilar membrane here in the cochlea. Here it might make sense to get an oval window. Kind of coming in. And this is the unwound snail. This should actually be going around 2.5 times, but here's the helicotrema up here. The round window down here. This cochlear duct is what we're going to focus on now. Which is the middle part. And this is where you have the hair cells. The hair cells are sitting on what's called the basilar membrane, so in which I need to put in here and talk about more specifically.
So the cochlear duct has the hair cells, or the organ of Corti has the hair cells?
They're both true. Organ of Corti has the hair cells and it's located within the cochlear duct. So the basilar membrane is kind of the floor for the hair cells. Then we're going to put the hair cells on top of that. Let me get a smaller. And then there's going to be another membrane, a little, they call the tectorial membrane on top of those. So, Ethan, the organ of Corti is this. Organ of Corti is the hair cells with that little membrane on it, the spiral organ of Corti. It includes the hair cells. So when people talk about the organ of Corti, it's like the hair cells and associated structures sitting on top of this basilar membrane. The basilar membrane is not homogeneous. It's not the same on the different sections. So there's this idea of mechanical resonance. If you take any object, I have a glass. It's just by its physical structure, it has a particular frequency it vibrates at. Right? It's due to the actual physical mechanics of the object that make it vibrate at that strings, like, here's a ukulele, right? I can, like, each string vibrates at a different frequency just naturally based on its thickness, it's how tight it is, right? So each string has its different thing, and it's based just on the physics of the mechanical structure. The basilar membrane is set up so when you are near the beginning here, near the round window or oval window, this part vibrates most at high frequencies. When you are up near the helicotrema, it's much kind of floppier, and this is going to have its resonant frequency at much lower frequencies. Just like, right, this would be like near the round and oval window part. It naturally vibrates at high frequencies. Up near the helicotrema, it just naturally vibrates at lower frequencies. And not only is that what it would vibrate at if you plucked it, it's the energy where it actually absorbs and starts vibrating itself. I don't know if you remember those, is it live or is it Memorex kind of commercials where, and the glass would like shatter. It's because she's singing, it's like, if you know what is the resonant frequency of this, and it's a very pure tone, and I sing at it exactly at that tone with enough energy, it'll just absorb it until it just like shatters, at least in a commercial. No, you can do it for real. So basically what's going to happen here in the cochlea is this. And we're in the cochlea. The sound, the vibration comes in, it's going up here. As the vibration goes up and down, depending on if it's high or low, it's going to be absorbed and shake the basilar membrane at a different section, depending on its frequency. If it's a low frequency, then the basilar membrane is going to vibrate more down on this part. If it's high, then it's going to vibrate on this part. Right? So depending on where the pitch is, it's going to vibrate, be absorbed and vibrate the basilar membrane on a different section. And then what's going to happen next as the basilar membrane starts vibrating?
The hairs are going to bend?
Exactly. You've got these hair cells sitting on top, kind of sandwiched between the basilar membrane and this tectorial membrane. They're going to wiggle and jiggle. The hair cells are going to bend and they'll send they'll send action potentials to the brain. That sound is happening and the brain will know the frequency of the sound based on where along the basilar membrane the message is coming from. Does that make sense?
So what exactly is the role of the tectorial membrane?
It's kind of to anchor the top of the hair bundle. So like when you jiggle its butt, the hair bundle on the top is going to bend.
But it's not sending the action potential?
No, the hair cell is sending the action potential.
Okay.
Or I should say the hair cell is talking to a neuron, which sends the action potential. The hair cell makes officially what's called a receptor potential, which then initiates a sensory neuron.
Okay, so
think about tectorial membrane is like if your hands were holding onto a rope up here and you're standing on a platform that then starts moving, it's going to make your arms bend because you're holding on here as your feet start jiggling. Does that make it more intuitive? Yeah. I'm sorry, Nikki, say that again.
So the basilar membrane vibrates the hairs, which sends the action potential to the brain, and it lets the brain know that like sound is happening and like the location.
Yeah, so the location tells it the frequency.
Oh, okay.
Right, but that is hearing. Hearing is basically air vibration is a stimulus, then vibrates your eardrum, which then is transmitted through the little bones, vibrates the oval window, sends that up the tympanic membrane, up the vestibular duct, down the tympanic duct. And in that process, the basilar membrane will start jiggling. Depending on the frequency, it'll be either up near the helicotrema for low or down here for high. When the basilar membrane jiggles, it jiggles the organ of Corti, which are the hair cells. And as those hair cells bend, it sends an electrical signal to the brain, and that's your experience of hearing. I
Can
should,
can I say it really quick just to make sure I have it right?
sure.
So when the sound or the vibration comes in through your oval window, the basal membrane jiggles or vibrates where the frequency is at, like the location of the frequency. And then that is going to move the organ of Corti, which contains your hair cells that send the receptor potential to the neuron to send an action potential to the brain.
That's basically it.
Cool. Thank you.
Yeah, I mean, the frequency is a the frequency that the basilar membrane vibrates at is just an intrinsic property of it. So I would say it vibrates the basilar membrane, and what part vibrates will depend on the frequency. This is also how cochlear implants work. When somebody gets a cochlear implant, they literally take an array of electrodes and just shove it up the cochlear duct here. And then you have little electrodes, again, all along the way. And then somebody has some little electronic thing that's listening to the sound outside and then analyzes it for frequency content. And depending on the frequency content of the sound will activate the hair cells on the appropriate area of the cochlea that would normally have been activated by a frequency or by sound with those collection of frequencies. Does that make sense? It's just artificially activating the hair cells or the nerves at the appropriate place along the cochlea, as if the system had been working properly. In general, it's not going to be nearly as subtle and detailed as actually having it work for real. People who get these, it helps them. It's not like it restores their hearing to where, normal hearing. Although I think kids who get this stuff really early, their system can actually kind of adapt around it and they can actually have pretty good hearing. But if you as an adult, it's going to help you, but it's not necessarily going to just fix your hearing and make it perfect or something. What else do I want to say about hearing? Other things. So the hair cells here, I'm talking about the hair cells. Are they afferent or efferent?
Afferent?
They're afferent, exactly. They are sending messages to the brain. It turns out that the ear, like if you just put a really small microphone in somebody's ear, you'll hear sounds coming out. They call them otoacoustic emissions. It turns out that there are also efferently controlled hair cells. They're called outer hair cells. So what happens, you have these other hair cells in here that their job is actually not to do sensory transduction. They actually are controlled by your brain and can push and pull. As they push and pull, they actually create vibrations in the fluid, which then goes backwards and then operates your eardrum as a little speaker cone. But when people was trying to figure out what's going on here, it turns out that the ear is more complicated than just a mechanical resonance of the basilar membrane to determine frequency. So I just want to put this in here that the actual thing going on in the ear is a little more complicated than just the basilar membrane is mechanically tuned to from high to low and will vibrate at a particular frequency, and that's the end of the story. There is also what we call these active components in there, where there are little motor hair cells that are altering the tuning properties in there. And part of the reason why this is important, I'll just bring it in very briefly. You know, when you think about the eyeball, you know, and you're looking at two dots, and they land on the retina, there's different rods and cones that are responding to those two different dots. So it's not surprising you can tell two different dots, because you have your independent little receptors that are responding to the different things out in the real world. With hearing, it's really different, right? You have an eardrum, and no matter whether it's a dog barking, woof, 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 or somebody yapping at you, blah, 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 or there's the alarm clock going off, ring, 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 and there's some guitars playing, all those sounds glom together, become one, just some total vibration that is then vibrating your eardrum and going into your inner ear. And yet, you still are typically able to tell, like, oh, there's somebody yapping and there's a dog barking and there's an alarm going at the same time. You know, you can discriminate and tease apart all this different stuff, even though you have one kind of big glom of vibration happening on your eardrum. You know, in order to do that, you need to be able to have very fine frequency as well as timing information preserved. So that's, and again, I'm looking at the clock and I could go on and on this stuff and why it's so important, but I will just say, you know, you're very good at determining frequency, like what is the speed of the vibration of the sound, but also the kind of the timing. Like, when did the sound start? When did it stop? For regular, just mechanical, like mechanically tuned resonances like this, it might, in fact, the purer the tone, the worse my timing information is. It just keeps, you kind of get lost. Like, you know what the pitch is, but you don't know even when it started or stopped, because it just keeps kind of vibrating. Your ear has these much more advanced filter or tuning properties where you can get very fine frequency information, and you can also know when the sound started and it stopped. And that, a big part of that is these active hair cells, these hair cells that are doing actually like kind of motor stuff within your ear. So just kind of putting that out there. The reality of the tuning in your ear is more complicated than just the mechanical resonance of the basilar membrane. So this is frequency and timing, or temporal information. Sometimes frequency is like spectral. Or, so I know I kind of moved through that fast. Are there any questions about that?
You said active components. What were those?
Those were those outer hair cells, the hair cells that are actually efferently controlled, that actually exert forces on the membranes in the ear. And it's where you have this kind of more fancy ability to do frequency and timing information. In fact, part of the reason why they knew to look for them was when they looked at the tuning properties of the ear, they realized that this is not something that you could get just from a passive kind of mechanical kind of filtering system. So that's when they knew, like, okay, there's something more complicated going on. And they realized, oh, some of these hair cells are actually not just turning movement into voltages. Other ones are being controlled electrically and moving. So there's this bidirectional thing going on. So what else? Again, I could spend lots more time talking about This is also really useful just for detecting signals from noise. Right, if you want to you have a tiger sneaking up on you, it's one little crack of a branch or rustle in the leaves in a whole forest where there's birds tweeting and wind blowing and trees rustling and stuff. And yet and again, that's one little bit of energy in this big sum total of all the vibration that's happening all at once. And if you have good ability to discern frequency and timing, it allows you to have a better chance of knowing that you got a tiger sneaking up on you. In fact, we're going to see the timing come up in just a few moments in our lab, in terms of just determining where in space is the sound coming from.